Welcome all to Part 1 in Developmental Biology and Medicine. In 2019, the United States reported an infant mortality rate of 5.6 deaths per 1,000 live births. Notably, birth defects accounted for over 20% of these deaths, while complications arising from prematurity contributed to another 20%. Collectively, this means that nearly half of all infant deaths are linked to developmental issues, underscoring the critical need for effective prevention and intervention strategies. Public health initiatives play a pivotal role in preventing birth defects. Genetic counseling and prenatal diagnosis empower families with information to make informed decisions. Primary prevention strategies, such as recommending folic acid supplementation, have markedly reduced neural tube defects. Additionally, campaigns focusing on avoiding teratogens, like alcohol during pregnancy, have been instrumental. Looking ahead, ongoing research into developmental processes holds promise for therapies that could further reduce the morbidity and mortality associated with birth defects. Dysmorphology is the study of congenital birth defects that alter the shape or form of one or more parts of the body. Medical geneticists are essential in diagnosing these defects, recommending further evaluations, and providing prognostic information. They develop management plans, explain the causes of malformations, and offer recurrence risk information to families. This collaborative approach, involving specialists in pediatric surgery, neurology, rehabilitation medicine, and allied health professions, ensures comprehensive care for children with serious birth defects. Birth defects are classified into three main types: malformations, deformations, and disruptions. Each category reflects different underlying causes and mechanisms, helping clinicians diagnose and manage these conditions. Malformations stem from intrinsic genetic issues affecting development. For example, Greig cephalopolysyndactyly syndrome, caused by GLI3 gene mutations, results in extra fingers or toes. Such malformations can sometimes affect multiple body parts due to shared developmental pathways. Deformations result from physical constraints during fetal development, especially in the second trimester. Arthrogryposis, or joint contractures, can arise in cases like twin pregnancies where space is limited. Many deformations are treatable with physical therapy or devices. Disruptions involve the loss of normal fetal tissue, often due to factors like trauma or amniotic bands. Amniotic band syndrome can result in limb amputations when bands constrict the fetus. These cases are more challenging to treat as they involve actual tissue loss. We will now discuss in further detail the different causes of malformations. Malformations can arise from chromosomal imbalances, with trisomies accounting for about 25% of cases. Small deletions or duplications, known as copy number variants, make up another 10%, while single gene mutations contribute around 20%. Each type of genetic cause provides insight into the underlying complexity of these birth defects. A significant proportion of malformations, about 40%, are multifactorial, involving both genetic predispositions and environmental factors. Another 5% are attributed to teratogens, substances that can cause malformations when exposure occurs during pregnancy. Pleiotropy describes how a single genetic mutation or environmental factor can impact multiple organs. Syndromes, like branchio-otorenal dysplasia result in parallel abnormalities, whereas sequences begin with one defect that leads to additional complication. Rubinstein-Taybi syndrome, a pleiotropic syndrome, is caused by mutations in transcriptional coactivators like CBP or EP300. The syndrome manifests as developmental delay, distinctive facial features, and broad thumbs, showing how one gene can have widespread effects on the body. Here are some images of the different physical characteristics of Rubinstein-Taybi syndrome. The Robin sequence is a developmental sequence where restricted growth of the mandible early in gestation causes the tongue to lie in a posterior position, obstructing the normal closure of the palatal shelves, which leads to a U-shaped cleft palate. This can occur in isolation or due to conditions like Stickler syndrome, where mutations in collagen genes affect multiple areas of development. Recognizing this as a sequence rather than a primary cleft palate malformation is essential, as it influences the prognosis and treatment for the child. The next few slides will serve as an introduction to developmental biology. Developmental biology is fundamental to medical genetics, as it helps explain how a single cell can give rise to a complex organism. This process, starting from a fertilized egg, involves the formation of trillions of cells and numerous distinct cell types, all in a predictable pattern. Understanding this transformation is essential for understanding congenital conditions and malformations. Developmental biology has evolved from simple observations in embryology to sophisticated molecular studies. Early embryologists used animal embryos to observe development, but now, with techniques like single-cell RNA sequencing, scientists can analyze gene expression patterns at each developmental stage, revealing the diversity of cell functions in both embryos and adults. In the next few slides, we will discuss the impact of the environment on our genes. Evolutionary biology distinguishes homologous structures, which share a common ancestry, from analogous structures, which evolved independently. For example, the arm of a human, the wing of a bird, and the wing of a bat are homologous. However, the wings of birds and bats are analogous, evolving separately to serve the same purpose. This concept of evolutionary conservation allows us to study human development using animal models. Genes provide the instructions for development through various products like transcription regulators and growth factors. Unlike a detailed blueprint, the genome guides processes such as growth and differentiation, resulting in the mature structure of tissues and organs. Developmental disorders often arise from genetic variants that disrupt these pathways. Genetic variants can influence development in a probable way. For instance, the disheveled 2 variant in mice leads to congenital heart defects in only half of the carriers. This effect highlights that genes regulate but do not fully control development, allowing for variation in outcomes. Environmental factors, or teratogens, can disrupt development by interfering with key pathways. Examples include thalidomide, which causes limb malformations, and alcohol, which primarily affects the central nervous system. Recognizing these teratogenic effects is critical for both public health and understanding developmental processes. Neural tube defects in detail. Neural tube defects, or NTDs, are among the most common and severe birth defects. Anencephaly and spina bifida are the primary types, affecting brain and spinal cord development. These defects contribute significantly to stillbirth and early infant mortality. A significant factor in neural tube defects is maternal folic acid deficiency, which is crucial for DNA synthesis. Deficiency during early pregnancy, especially in mothers with an MTHFR gene variant, increases the risk of NTDs, likely due to metabolic disruptions involving homocysteine. Genetics also play a role in NTDs. The MTHFR gene variant is common and can reduce folate metabolism efficiency. Mothers homozygous for this variant are twice as likely to have children with NTDs, illustrating the genetic susceptibility to these defects. NTD prevention includes folic acid supplementation, which has reduced incidence by 75%. Prenatal screening with maternal serum AFP levels and ultrasound helps identify at-risk pregnancies, enabling early intervention and management. Public health initiatives, such as mandatory folic acid fortification, have reduced neural tube defect rates by 35% in the United States. Continuing education and screening aim to lower these rates further, making a significant impact on public health. The end. Please note that this was an AI-generated voice.